Thank you for coming. So how many of you are here as a part of Hub Week? All right, great. Thank you so much for coming. In honor of you and all Hub Week participants, I am obligated to share a few words from our sponsor, which I will do by reading off the sheet, no shame, full transparency. So Science in the News is very proud to be a part of Hub Week, Greater Boston's Festival for the Future. Hub Week celebrates innovation and creativity at the intersections of art, science, and technology. We believe that the future is being built here in Greater Boston. And Hub Week is, of course, all about collaboration. So this year there are over 180 different organizations, including Science in the News, that are a part of Hub Week. And we're showcasing an incredibly diverse array of different works. So I'm glad that you chose this lecture tonight among the many options that you had. And so in addition to our fall lecture series, which does occur every week on Wednesdays here at HMS during the fall semester, Hub Week is also going to be doing a centralized festival in the City Hall Plaza. And this is going to be called the Hub. So it's going to be open starting tomorrow, the 12th, through Sunday, the 15th. And there's going to be events, art installations, and it's going to be a very diverse, multi-idea festival. And of course, there will be live arts being performed at the festival at all times. And so if you're curious about it, all this, and haven't yet taken the opportunity to memorize everything on the Hub Week schedule, no worries. You can still go to hubweek.org for a full list of events. And if you're tweeting or whatever it is kids do these days, use the hashtag Hub Week. Check out what's going on. You can also follow them on Twitter, and they'll be posting like crazy the next few days. Yes, our unique code. Ready? 36391. If you use that at registration, you'll get super cool discounts, and then give us the credit because we're great and we got you that code. So thank you for being here and being a part of Hub Week. And since many of you, I realize this may be your first Science in the News lecture, I just want to give a very, very quick rundown on what Science in the News is all about. I've already used that phrase many times, and you're probably wondering what are you guys doing. So we're a graduate student organization here at Harvard. We put on free events and write articles and create science media for the general public because we believe that there is an enormous gap in science communication that we are hoping to fill by training the next generation of scientists to better communicate their work. The Fall Lecture Series is one of our cornerstone series of events. We've been doing this for over 18 years, so Science in the News is now legally able to vote. And that's very exciting for us. So we do this, as I said, every Wednesday here at HMS. In the fall and in the spring, we do a similar series, again, on Wednesday evenings, but then it's every other week at the Harvard Cambridge campus. So if you're curious about our events, I encourage you to grab our green flyer here at the front. This is our flyer for the fall lecture series. There are going to be 5 more lectures in the series for the remainder of 2017. It's still 2017. It is. I also want to quickly advertise one of our other event series called Science by the Night. This one is 21 and over, but this is a science cafe series that happens at the Burren and Aeronaut Brewery in Somerville. So if you're curious about hearing about a local faculty member's scientific work and getting the chance to chat with some of their lab members over a beer and free fries, I highly encourage you to check out this event. If you don't feel like leaving the apartment, that's okay too. You can find a ton of articles, archived video lectures, podcasts, art, and general graduate student ramblings at sitinboston.com. And I think I've said more than enough. I don't want to keep you too long for the lecture, but Dana's going to take you really quickly through how this will work tonight.
Yeah. So hi, I'm Dana. I am the other co-director of Science in the News along with Katie. And tonight's lecture, you're welcome, this is Katie. Tonight's lecture is going to be by some of those graduate student scientists that we are helping train to communicate their science. And they're going to be giving you a lecture in 3 parts. And in between each part, we'll have a break for questions. So we ask that you please hold your questions until those breaks, and we'll make sure we have time to get to all of those. We'll also take a brief intermission after the second question break, so you can get some more snacks or ask some one-on-one questions as you so desire. Before we get started, I wanted to remind you guys that And along with our Science in the News flyers, the colorful ones up here, we have a handout about tonight's lecture that has some information about our speakers, a description of what you're about to hear about, as well as some terms that you might want to find, or some resources where you can learn more about the topic. So feel free to pick one of those up either now, during the intermission, or on your way out. And also we have some paper surveys up here to get your feedback about the lecture, about our performance as Science in the News, about the lecturers' performance as lecturers. So since we are a training organization and we are all students and we are still learning, we would really appreciate your feedback, especially if this is one of your first events with us. So please, during intermission would be a perfect time. Come down, grab a survey, and just fill it out really quickly and let us know what you think. All right, without any further ado,
We're here about GMOs.
Yeah, here we go.
Okay, guys. All right. Well, I'm Allie.
I'm Max.
And we're both second-year PhD students. We're working together in the lab on a research project to develop bacteria with new genetic code. And today we're going to give you a whirlwind tour of GMOs.
So, genetic modifications are a really broad concept. If you take a walk in Central America looking for wild corn, this is what you'll find. And through thousands of years of growing a tasty citrus crop, we have what we know today. At the same time, once we began to understand DNA better about 50 years ago or so, we found new ways to make changes to our food. And surprisingly, most of the food we eat today has some amount of modification. But what is it about what on the right and what on the left that is different? Genetic science doesn't just top headlines, but it can save lives. Insulin is an important gene for diabetes, but before laboratory methods, we had to use insulin for things. Now, insulin identical to that in our bodies can be provided for a fraction of the cost. Genetic changes aren't always hard to see. The flashy stuff, or even the glowy stuff, captures at least our imagination. As the cost to make these modifications continues to get smaller, what does that mean for the future? So we're happy you've come to share some time with us so we can share with you a look at just what these things are, what technology binds them together, and what we need to think about beyond just the testing. So if we're going to take a look inside of GMOs, we need to understand what it is we're looking at. So our first section will be a quick crash course on what does DNA actually do, what are genes, what do proteins have to do with genetics, and questions. So life can look really different and that's really exciting, from bacteria to bugs, from trees to turtles. And one of the best ways to understand something large and complicated is to look for patterns. So let's be biologists together and ask ourselves, what does all life have in common? So let's start by what we can see. All life on Earth starts from something small and it grows bigger. It gathers energy, it finds food of some kind. It reproduces to make more of itself. It reacts to its surroundings in some way or another. And over generations and through long amounts of times, it evolves from one form to another. So what is common under the hood here? Through the last century, and especially the last 50 years of biology, biology has uncovered a pattern inside of all life that can help anchor us to understand it better. Life is made of compartments, and inside of each of those compartments are instructions. Each of those instructions help build molecules. Those molecules come together millions of ways to create the abilities that we just looked at. So let's look at this, what this means in terms of molecules, and we'll better understand what genetic modifications are. So the cell is the unit of life. It's the smallest thing that does all of those abilities that we just saw. Here is an onion cell, here are some brain cells, and here are bacteria. Those are organisms with just one cell. At this scale, those tiniest dots might hardly be one bacteria. Everything inside of these cells are just molecules, though. They're not really alive. They don't do anything on their own. They come together. So how is it those parts inside of the cell come together and give it those abilities? What give those parts the instructions that they need? So as we look closer, one thing that our eyes land on is DNA. We find it in every cell. It looks like this, two molecules wrapped around each other. And if we look for patterns, we can notice one part of it is that it's made of 4 repeating units. And on top of that, each of those units is paired up in one molecule with its partner on the other. And so we can simplify the molecule even further into just a list of which units are in what order. We'll call these letters that the real name will be. So the whole molecule can be summarized to just the order of those letters. And DNA serves as an instruction for the cell, but what exactly are those instructions? So we might think that DNA holds the codes for traits or bigger abilities, but at its fundamental core, they hold the code for proteins. Proteins largely float on their own, so if the cell knows the sequence of building blocks how to make a protein, then it can build it. So a typical protein might look something like this. This is part of what your hemoglobin looks like. That's inside of your blood. You have your red blood cells. Inside of every blood cell is hemoglobin. And the shape and exact chemical properties of this molecule help it carry oxygen around your blood to different parts of your body. So it looks complicated, but how can DNA tell us how to make it? If we look deeper inside of it to see sort of its skeleton, its most basic form, we can see that it's made of two chains that are coiled together and then fold into a larger 3D chain. So on the left is surface, and on the right is really the core connections between the different parts of the inside. Each of those chains is made out of small building blocks called amino acids. And DNA is also made out of a chain of different molecules. So here, at the right time and at the right place, a small factory inside of the cell called the ribosome basically interprets certain parts of the DNA in little groups of 3 letters at a time. And it takes that information, and each of those specific little triplets of letters tells it which building block of the protein it should connect one after another. And when those building blocks are connected together, the protein largely automatically folds into something as complex and useful as hemoglobin. So there may be thousands of different sequences inside of the cell that tell how to make each and every one of these proteins. So hemoglobin is nice. It finds oxygen, and sometimes it lets it go. How does that create life? Before I say that, I just want to quickly define what is a gene and what is the difference in DNA exactly. DNA is more like paper and ink. It's the physical material and molecule that stores the information. Whereas the concept of gene is closer to something like a word. It's information that is stored inside. Specifically, that order of building blocks for the protein and a bit of information about where and when and what part of your body and what conditions you should make that protein. Depending on what DNA you got from your parents when you were born, you'll have slightly different genes written in. Those genes may be slightly different. Those differences mean the proteins in your cells may be slightly different, or they might interact slightly different. So you grow up differently than the person sitting next to you. On average, of all your DNA, you share 99.5% of the DNA with any other person. There is a lot of DNA in your cells. There's a full copy of pretty much every single one. 3 billion letters, so times 2, one copy is 3 billion from each parent. Tens of thousands of genes, and in smaller organisms like bacteria, there may be many fewer. Because they can do fewer things, so they're only So here's a drawing of what it looks like inside of the bacteria. Quite scientifically, at least trying to be very accurate drawing. If you could cut it down the middle, slice it open, and see all of the molecules, you can immediately see how crowded it is, and it's not really a big empty space with the molecule floating here and another there. It's really jam-packed. Here in green on the outside is the membrane that holds it together, and those long things, the flagella, they're kind of like little pellets that whip around and let the bacteria move in its environment, search for food, harness energy. So let's look inside. So this is pretty close to a depiction of what it would be like inside of a cell if you could see every molecule. On the outside in green, molecules are on the outside. Some proteins act as doors, those sort of column things, letting certain things in and out of the cell. You see the base of the flagella. In blue, there are all kinds of different proteins. Most of them might be enzymes, breaking down molecules, building up new molecules, taking in food, extracting things, using vitamins, using energy. In purple, you see those little factories I mentioned, the ribosomes. And they're creating in pink those little protein chains that haven't yet folded up into an actual useful protein that could be used somewhere else in the cell. And in yellow is the DNA. It's little bumpy structures. Each little bump along the rope of DNA is one pair of those little letters, storing all of the information to make the genes. And in orange around it proteins are attending to the DNA, protecting it, preparing it, controlling when each part is used, and so on. The DNA stays largely the same in the cell, whereas a lot of the protein is constantly recycled. It's broken down, new ones are made as they're damaged, and so on. And like a city, it's the millions and millions of interactions between each part that really make it work. Together, it's alive. So, what's really fascinating about this, and that we learned as we looked closer at these things in decades, is that the DNA, again, is just these four little subunits. And not only that, but if you took the DNA from that E. coli cell, and you took some from me, you put them side by side, they would look the same. And it's only that sequence of letters that's in a different order. This is true of every single thing on the planet. Same goes for proteins. If you take any two proteins, you untangle them, you could find that they were, at least initially, constructed using just 20 different building blocks. So this commonality is useful to us as scientists. And for lots of research, we can use with a lot of organisms for research into health. You can take bacteria and study them. They're easy to grow and learn about DNA in that context, and then see what applies to us, to human health, and so on. So you can connect all the different things we learn from different fields of research. And why you're here is it's an important question for bioengineering. Can we change something in the DNA, which would change how the protein acts, which would change perhaps something about what the organism does if that protein has an important job. And that is what value. Anyways.
Just on a size level, what are the size?
Largely, so the question is, what is the size of many of these different things in terms of different kinds of mediums? Animal cells and plant cells are a lot bigger than bacteria, and viruses might be even smaller than bacteria. Hundreds of micrometers, a micrometer is a millionth of a meter, might be the size of a human cell. A bacteria might be a couple micrometers big. You saw kind of the size between DNA, which is about 10 nanometers wide, just a couple atoms, but it's packed really densely inside of those. So in a human cell, a couple hundred micrometers across, but 6 feet length of DNA is coiled up inside of every single one of your cells. So a lot of interesting questions.
So it's very small. That's hard to imagine. The measurement term you use doesn't make any sense to me.
Sure.
But your body is composed of how many cells?
On the order of trillions. So trillions is from thousands to millions. Each of these is 1,000 times bigger. Then to billions, and then a trillion from that.
I think it's about 34 trillion, is the guess.
Yeah. So we've got a lot of cells, whereas the bacteria is just a single cell. Whereas in our bodies, each of our cells could have a different job. So DNA, instructions on how to build the proteins. Proteins all work together in different ways to generate the abilities that give the entire thing together the ability to live. And if you change the DNA, you might be able to change what. Oh, do you have another?
All right. So now that we know kind of the basics of how DNA goes to a protein, we're going to actually look at how we go about changing them. So genetic engineering relies pretty heavily on the fact that DNA goes as converted to proteins. While it's difficult to change a lot of proteins in the cell, there's only 1 or 2 copies of your DNA usually, and it influences not only what proteins are made, but when they are made and how much of them are made. DNA is very fragile and easily damaged. These damages to the DNA are called mutations. These can occur naturally, and they happen every time a cell splits into 2 new cells. Although cells do a very good job of fixing mutations on their own, some slip through, and the effects of these mutations may be seen in the proteins. So there are a lot of diseases that are caused by mutations in single genes. Huntington's and sickle cell anemia are just a couple of examples of these. Huntington's disease is a neurodegenerative disease that's caused by a mutation in the Huntington gene. Symptoms of Huntington's disease include loss of coordination, general loss of ability to do tasks like eating and speaking, making involuntary movements, and increased risk of mental illness. Patients usually start showing signs of disease around 30, and their lifespan is substantially lower. Although you can find out because it's a mutation whether or not you have the disease, there isn't a cure, and you can only manage symptoms as the disease progresses. Additionally, sickle cell anemia is caused by a single mutation in hemoglobin, which Matt showed previously. This leads to having red blood cells that, instead of being kind of nice and round and donut shaped, they're instead kind of a thinner, smooth crescent shape. These wind up getting kind of caught up in your blood vessels around your body, which ultimately leads to some organs not getting enough blood, which causes them to start becoming damaged, and ultimately can lead them to die. This means that a lot of patients wind up having pain, especially as the disease progresses, and often they don't live past 40 to 60. But are DNA mutations always this bad? So we're going to look at how mutations affect an organism looking at strawberries. Well, the strawberry we see here is kind of a decent looking strawberry. It's maybe not the strawberry you would pick immediately out of the box. So let's look to see whether genetics can give us a juicier strawberry in the next generation of strawberries, which is a very important point to make here. Is what we'd be looking at doing is mutating the plant that makes strawberries so we can breed it for the next generation, but mutating the strawberry in and of itself and not breeding it isn't going to cause any changes to the strawberry. So I kind of gave away the punchline in the last slide. So if the mutation of if we create a mutation in a gene that makes the strawberry juicier, the next generation of strawberries are going to be a lot juicier. Whereas if we make a mutation in a gene that doesn't really affect how the strawberry looks, we're going to wind up with a strawberry that looks exactly the same as our previous strawberry. Meanwhile, if this mutation is in something that actually makes the strawberry less juicy or causes it to not grow quite as well, we're going to wind up with a smaller strawberry than we previously had. And finally, if the mutation is in something that is super important to the strawberry plant to even make strawberries, we're not going to see any strawberries in the next generation. So, given mutations can have such a large impact on organisms as a whole, and we know that mutations in genes change proteins, we would want to look at the different kinds of mutations that could change the protein. So, we're going to look at 4 general kinds of DNA mutations, each of which could have a small or large effect on the protein made by the gene. DNA mutations generally change the shape of the proteins you get in the end. A protein acts as a molecular machine. It does some job, and changing the shape of the machine is going to change how well it does the job. In this example, we're going to kind of consider the protein like a machine you see around Boston Lab, which is a bike. So the first kind of mutation we're going to look at is a point mutation. This occurs when only a single letter in the DNA is changed. This is actually pretty important because this is the only mutation we're going to look at that has a very specific size that gets changed in the mutation. Every other mutation we'll look at could be changing from 1 or 2 letters to chunks of DNA that include multiple genes. So in our point mutation shown here, we change a G to a C. In our bike example, this can be as small as the person who puts it together forgets to add the screw that keeps the light on. You lose the light, but you still have a good bike. Whereas if the person forgets the screw that keeps the wheel on, you're in slightly more trouble. So another kind of mutation is a duplication. This occurs when the DNA accidentally gets a second copy of some part of itself. Here, in our example above, we now have 2 copies of CT. In our bike, we can wind up with something like a second light, which doesn't really change how the bike works, or could be advantageous depending on how you look at it. Or we could wind up with a second pedal on the same side of the bike, in which case you can't actually pedal the bike. Another kind of mutation is a deletion. This is when part of the DNA is removed. In this example above, we've lost our G and G. In our bike, this can be as small as losing a kind of tiny chunk of the handle, which still leaves the bike usable, or as large as losing an entire wheel. Our final kind of mutations are inversions. This happens when the DNA flip-flops and the sequence is now backwards. In our example above, we now have our GGCT has now changed to TCGG. In the bike, this can be as small as we reverse the direction of the wheel, but it still works if it doesn't matter what direction you put the wheel on. Or as big as changing the orientation of the bike seat's position. So just because we know how a mutation will change just the protein
doesn't mean we know how this change will affect the organism. Proteins don't act alone, but usually proteins don't act alone, but they act with other proteins. Changing some proteins will have more of an effect on the organism than changing others, but this can be pretty hard to guess. This makes it harder to change some traits of an organism than others. Many characteristics of an organism have a lot of genes that make it what it is. A juicy strawberry isn't necessarily juicy because of just one gene, but it could be juicy because of hundreds of genes, all of which contribute to making the strawberry juicier. Many characteristics aren't just influenced by DNA, but they also depend on the organism's surroundings. If my strawberry had the right mutations to become a juicy strawberry, but I grew it in the cold or I forgot to water it, I may not see a juicy strawberry. So we talked a lot about mutations, and we know that some mutations can lead to living things having different properties than others in their group. So some of these properties might allow an organism to survive for longer and have more children. As an organism with a certain trait is able to live to have more children, the mutation that leads to this trait is going to be passed down to other children until over time it's going to make up the traits that will be present in the majority of the population. This is what's known as evolution by natural selection. Here, nature is making organisms better able to survive by allowing certain individuals to live longer and to have more children. In the case of the finches on the Galapagos Islands, which we see here, one ancestor bird evolved over time into many different bird species of different sizes and beak shapes. This allowed birds to take advantage of different food sources that were on the island and reduce a lot of the competition between the birds for food. So if you look at the bird that's furthest away from me, that bird's a massive beak and it might be better at cracking open nuts. Whereas the bird closest to me has a pretty tiny beak and it might be better to kind of scavenge seeds around on the ground. So, but there are some traits that people want in animals and plants they interact with and eat that just aren't good to have in nature. Being a juicy strawberry isn't fantastic in the wild, but humans want to eat juicy strawberries. This is where artificial selection comes into play. Artificial selection also takes advantage of evolution. It allows humans to selectively breed those animals and plants that contain traits that are more useful to them. If you are out gathering berries and you see one plant that's producing juicy strawberries, you might mate it to another plant that's also producing juicy strawberries and breed over time, over many generations, to gradually get a lot more plants that are producing juicy strawberries. Humans have harnessed evolution since prehistory. For over 12,000 years, we've been breeding for cows with better milk production, for juicier berries, for more vigorous crop growth. We've been doing this long before people even knew what proteins or DNA were. So to overview, natural and artificial selection both rely on the same property of evolution by mutation. However, natural selection is selecting for those mutations that produce traits that are more beneficial for an organism to survive in nature. While artificial selection is selecting only for those mutations that give traits that are from plants or animals. But neither of these is particularly fast, and both only allow you to select for traits that are already in the organism. You can't select for a strawberry that makes a toxin to kill a worm that eats it if it's not already present in the strawberry. So what's the solution to that issue? The solution is why you're all here. Genetically modified organisms are just living things whose DNA has been modified using laboratory techniques. These changes generally occur before the organism is fully grown, as we'll see why later on. So there's tons of GMOs already, and they're all very useful. Or they are very useful, not all of them. We can make corn that grows more rapidly and isn't killed by certain bugs, but we can also make golden rice, which contains a molecule that the body needs to make vitamin A. This is really important, especially in developing countries where vitamin A deficiencies are very common. So, if GMOs are so great, how would I go about making them? So, we're going to look at an example in which we want to make insulin. Insulin is a very important medicine for diabetes. However, unlike most medicines, insulin is actually a protein and can't be made without an organism because we need DNA to make protein. So if we want to make insulin cheaply, we might want to get bacteria to make insulin for us. To do this, the bacteria needs to have a gene to make insulin in its DNA so that it has the instructions to make it. So let's say I'm a second-year PhD student with a tube of bacteria, and I want to make bacteria make insulin. There are certain steps I can go through to do this. First of all, I would go through articles and talk to other scientists to see if there's a gene out there that allows cells to make insulin when it's present in their DNA. As it turns out, there is. If a cell has the insulin gene, it's able to make insulin, as seen by these kind of little puffy purple clouds, which are used to represent insulin, and the insulin gene as the green bar inside the orange dot. However, any cell without the insulin gene isn't going to make insulin. I'm going to take a copy of the, if this is the case, I'm going to take a copy of the insulin gene out of the cell that already has the insulin gene in its DNA. But one copy of an insulin gene isn't really going to help me make a genetically modified organism. If we're trying to get a gene into a cell, having only one copy of your gene gives you pretty low odds of it going into your cell versus a lot. So instead of having just one copy of the gene, I'm going to copy it a lot using laboratory techniques and make tons of copies of it. I'm going to then mix these copies of the insulin gene with my bacteria, shown here with the reddish colored DNA. Electrocute this mixture, which is going to open holes in the bacteria, and while opening holes in the bacteria, it is going to kill most of the bacteria. The holes, I'm hoping for bacteria where the holes are just large enough to allow my gene to slip inside. But after all that, my bacteria isn't actually making insulin. And why might that be? So cells don't like DNA that isn't there. So when they find DNA within them that they don't recognize, they're going to get rid of it. In order to get my gene to go into the cell, into the bacteria cell, and have it recognize it as part of the bacteria, I'm going to need to disguise my gene with DNA from the bacteria. Here I've shown it as the kind of reddish color attached to both ends of my gene. If I'm careful about picking the bacteria's DNA that I disguise my gene with, the bacteria is going to add it to its own DNA. I'm now going to repeat exactly what I did before, but I'm now going to use my disguise gene. I'm going to copy it a bunch of times, mix it with my bacteria, electrocute the mixture. Holes are going to open in the bacteria. And if I'm lucky, my gene is going to slip in. And now, because the bacteria thinks the gene is from its own DNA, because I've disguised it, it's going to put it into its own DNA. We have now engineered bacteria that makes insulin. This is fantastic, because if we do this with the human insulin gene, we now have a ready supply of human insulin for someone who has diabetes. So mutations affect bacteria and animals differently. Most bacteria have only one copy of their DNA. So if our bacteria's DNA is modified, any new bacteria it creates is going to get the mutation. Oh, my modified DNA is showing. So, however, in humans, how a mutation affects the body depends largely on the stage in development in which the mutation occurs. When you're first conceived, you start off as a single cell. If you get a mutation at this point, every cell in your body is going to have the mutation. However, if one of your cells gets a mutation later in development, say when you're like 8 cells large, only the parts of the body that that cell divides into are going to be affected. If the mutated cell leads to the heart, your heart's going to contain a mutation, but something like your brain isn't going to. Meanwhile, if you're 30 years old and one of your skin cells gets a mutation, only that skin cell will have that mutation. So, given that's the case, how would I go about putting DNA into an animal cell? There are many ways to do this. However, we can't easily electrocute animal cells. So we have to rely on something that you and I all have in us right now. And the answer to that is viruses. So viruses are extremely good at putting DNA into other cells. That's actually their only job is to make more of themselves by doing this. So some viruses are happy to put their DNA into all kinds of cells, whereas some viruses only want to do this in things like your nerves or in your blood. For our example here, our virus is going to be pretty happy putting its DNA into whatever animal cells we give it. So if I start with a normal virus, with its DNA shown in red in this diagram, and I mix it with my animal cell, the virus is going to grab onto the cell and inject its DNA into the cell. Like we tricked our bacteria before to take up our insulin gene, the virus has ways of tricking the cell into thinking that its DNA is the cell's DNA. This is going to cause the cell to make a lot of viruses using the virus's DNA as a template. Eventually, the cell is going to make so many viruses that the cell is going to explode and release viruses everywhere. And these viruses will do exactly the same thing as their parent virus, and so on. But we don't want to make tons of viruses to do genetic engineering. We just want to get our gene into an animal cell. So is there some way that we can get a virus to just do that? So if I take all the virus DNA out of its body, it's now completely empty. The virus is still going to want to do what it always does. It's still going to grab onto the cell. But it doesn't have any DNA to put into the cell, so the cell isn't going to be told to make a lot of viruses. The cell is just going to continue to do what it always does. Removing the virus's DNA from its body makes it safe to put other DNA into the virus. Removing the virus's DNA from the virus's body makes it safe to put other DNA into a cell without the risk of getting more viruses. In this case, the viruses are just a delivery system that cannot multiply when treated this way. So, for my example of putting a gene into an animal cell, a lot of animal cells already make insulin, so we're not going to get much out of adding another insulin gene to an animal cell. Instead, I'm going to take pig cells immediately after fertilization and give them the instructions to make a protein, which I'm going to call green, that glows green. In this case, I'm going to get pigs that glow green. So we learned from engineering bacteria to make insulin that we have to disguise our gene with DNA from the cell we're putting it into. Here, I've attached pig's DNA in blue to either side of my green gene. So I put my disguised gene into a virus again, which in turn delivers the disguised gene into the pig cell where it's incorporated into the pig's DNA, which gives us 3 pigs, which is our different set. So if we talk about genetic engineering, we can't possibly have this discussion without addressing CRISPR. CRISPR gene editing allows us, CRISPR gene editing uses proteins that the bacteria uses to defend itself against viruses by cutting any virus DNA inside of the cell. However, scientists have manipulated it a little so that it now edits genes. This has allowed scientists to fix disease-causing mutations in some cells and to change how much protein a certain gene will make in plants. I would like to be clear now that CRISPR is not an in-practice therapeutic, and a doctor can't prescribe it to you. I don't actually use CRISPR in my research, but Max over here does. And you're now ready to introduce.
Alright, yeah, questions?
I have 2 questions. One is, you are talking about the insulin, how is like we can do bacteria to do it. And I was wondering why we don't give it to the people, and instead of like modifying the bacteria, we just modify whatever is insulin produced. And you say that because it's more complex and things like that. So right now there is like for example, a medicine for leukemia when they can change the white cells, I think so, the T cells, and then this called against the cancer. So what is the difference? Because the immunotherapy can work for the whole body, but they don't fix the insulin production. That is one question. And the second question is...
I just want to answer the first question first, and that way I'll
We summarize it.
Yeah. So the question is, if we can move genes between different animals or different organisms, why do we put the insulin gene into bacteria? Whereas in the recent research event, there were changes to human white blood cells in fighting cancer. And so in the particular example of insulin, the reason insulin is injected is not because our pancreas cannot make the insulin. It's often either because our own cells immune cells are attacking the pancreas cells themselves, so they're just not even there anymore. It's type 1 diabetes. And in type 2 diabetes, a lot of the other cells don't respond to the small amount of insulin that the pancreas makes on its own. Like you type 2. So in this case, it's about how you treat with insulin, and the issue isn't that we can't make it on our own. So it's about the diabetes itself, and not so much that our own cells can't make it. Whereas in the T cell example, I haven't read the details of that one, but there I believe they could. There are proteins on the T cells that help them identify different, different cancers basically. Some T cells fight bacteria and viruses, and others can recognize when a cell becomes cancerous. The cell will actually put little flags on the outside and say, "Help! Something's wrong." And so, a lot of these therapies, maybe not explicitly, but they make edits so that those white blood cells can attack that better. So there's teaching those cells.
Sorry, I will append onto that briefly that they are trying to work on gene therapy for type 1 diabetes in humans. I think a study came out within the last 2 years where they've actually genetically engineered rats to produce insulin and successfully treated, rat diabetes. So it is something that they're working on, but there definitely are complications that don't make it a perfect equivalent to the T cell therapy that you mentioned.
Second question. So I was reading that or watching a video, and they say that with the CRISPR, they were taking in some spermatozoids and some eggs, and they modified with the CRISPR some spermatozoids, like an egg or something like that. And when they made the spermatozoid with the egg, which is female, I guess, it fixed all the mutations made by CRISPR. And so some people were wondering like if CRISPR is going to work properly because when you change something, it goes to the egg, so the egg will fix everything like a reset all the change made by CRISPR. So I don't know if it's true or.
Yeah, the question is, you read, so I have to repeat it for the the question was basically CRISPR was introduced into sperm and egg cells, and together when they combined, a lot of edits in that DNA were changed as you read. I would say that could be possible, I think. One of the big issues, again, that we mentioned with the strawberry example is it's often hard to understand what to fix when for different diseases or things like that. In the term like Huntington's disease, you can fix the one gene that we've learned from before is the one problem. But a lot of other things, there are hundreds of genes that are all working together. It can be very difficult to understand why exactly is it changing. You've heard maybe of protein folding is a computational problem. And that's because even if you know change this little sequence of amino acids, it can be hard to understand how they folded these things, then how that will affect other ones. So sometimes these things are clinically possible, but you don't always know if it will fix or change whatever you do. So I'm not as familiar with that example, but it's kind of broad.
This might be a dumb question, but how can you remove DNA from a virus?
So the question is, how can you remove DNA from a virus?
It's been a long time since I've learned about viruses, but basically a virus, what it does is it has basically all those proteins on the outside, and it has DNA on the inside, and some other enzymes that can cut our DNA and tries to sneak its DNA into our cells, so that we just go over it thinking it's hemoglobin or something, and then in the end it makes all those proteins, and together those come together and build inside of our cells more viruses, and sometimes those can get released. I'll give one guess as to how it could be. I'm sure there are lots of different ways that it could be removed. But if you made a virus that were missing some of those enzymes that it needed to fold itself together, to pack all those different virus pieces together, then what would be released could be basically empty virus particles. It would just be the outside that connects to something and then wouldn't have what's inside. But that could be one example.
I think I have maybe a little more to add to that as well, and I'm not 100% sure about this because it has also been a long time since I've learned about this, but usually it's not a completely empty virus because otherwise they wouldn't be able to make, their like protein coding and make their little legs and make the injection needle and all the stuff that they need to actually put genetic material into a cell. But they just, I think exactly as Max said, they get rid of the things that are making viral specific proteins. So they're getting rid of all the virus specific stuff and just keeping the packaging. So like repurposing a box from Amazon.
Some of the proteins in a virus are used to copy back the DNA that it inserted into us, because when it makes another virus, it needs the copy of instructions for the next one. So if you get rid of those packing materials, it can't.
How many NGOs do you
Yeah, how close are you to going from the amino acid string to calculate what the shape of the protein?
It's the question. The question is, how close are we from just that string of this amino acid to a full piece? I actually tried to do that for a project when I joined and it's really difficult. The short answer is it gets exponentially harder the more amino acids you're trying to do. So a standard computer, or maybe like a supercomputer you could borrow at the universe or something, could like do a couple dozen, a couple hundred even of amino acids. And after that, there are just too many. You know, one amino acid can interact with every single other one. The next one can also interact with every single other. It has to basically simulate all of the attraction between each of those, and it simulates the interaction with the water around it, other proteins, all these things. So when scientists understand the structure of proteins, they can look at it in other ways, like X-ray crystallography, where it basically crystallizes lots of proteins, shines X-rays through it, and looks at kind of the shadow of that. And from that can reconstitute what a protein might look like. So you can do that for really big ones if you have the protein already. And sometimes those kind of mutations can use that as a reference model, and they say, okay, this part of the protein will probably fold just like this, but this part will be different, so they can estimate it and make guesses as to that. But for now, to give you a number, probably a couple hundred. And there are proteins that are 2,000 or 3,000 long. And those can combine into bigger complexes. That hemoglobin I showed you, hemoglobin is made of those two chains, but that combination of two chains actually shows up kind of like a 4D clover. And it's those four complexes together that, so there's a lot of data.
Something that's pretty fantastic about how science has progressed over the, I don't know, even 20 years, is that now you can even just look up a protein and find its DNA, or a protein find its amino acid sequence. There's predictions and sometimes even images about how it folds. You can find all sorts of this just online or searching for protein.
So we can take a short intermission if you want to grab some snacks or something. At the end of our talk, sorry, we're done with part 1 and 2, so we just have a part 3. At the end of our part 3, we like to share a short lab tour, just a quick walkthrough of the space that we work in day to day. That's across the street in the big shiny blue R&D building. So we have a little sign-up sheet for the first 10 if you'd like to join us for that afterwards. It shouldn't take too long. And then we'll be back in maybe 5 or 6 minutes, and we'll be here for questions.
And really quickly, while I still have your attention, I do want to put in another quick plug for our surveys. Again, this really helps us grow as humans. So if you have a moment, please take some time to fill out our surveys before we leave the lecture. Thank you.
What would be the topic in part 3?
So the topic in part 3 is looking at it so that you might
Yeah.
No, I mean like
Okay, I'll talk to you.
Yeah, so there's a lot of different ways of trying to one of the constant considerations is this means that anything that's like has a certain like basically certain physics. Anything smaller than that can't form can't make a picture of that. So there are biology where you flash rays cells slowly all this kind of down to the all of these cells that might basically crystallized with round crystal. That's all, yeah.
Yeah, so I'm not sure how long we'll see that everywhere.
Probably get it to me.
That was the first one time. And plus, Pharaoh switched on his own.
All right, everyone, we're going to get started again. All right, so now that we talked about how we would actually do genetic engineering, it's pretty important to talk about the ethics behind genetic engineering. So throughout part 2, we talked about different things that genetic engineering can do, from making green cakes to making insulin-producing bacteria. We're facing a lot of issues as a society right now. It's estimated that there's going to be an additional 3 billion people on the planet by 2050. These people are all going to need to be fed. Current issues resulting from human use of land, such as pollution, soil erosion, and climate change, decrease the amounts of farmland that's available. Farming also has a lot of resources that it uses, including water, fuel, chemicals, and many, many more, to care for a large number of plants and animals. So consider the following. So artificial selection for high milk production cows uses a lot more resources than you might think. If we start with one heifer, we have to provide her with food, medicine, water throughout her entire life. Once she is ready to be bred, we also have to provide her with a bull to be bred with. Then we have to continue providing her with food, water, and medicine through her entire pregnancy. And only then, after she gives birth to a calf and becomes a cow, do we find out whether or not she's a good milk producer. Because cows can only make milk after they have a calf. So if she is a good milk producer, and I can vouch for that cow being a great milk producer, I then want to select for other good milk producers from her offspring to breed to kind of keep up the milk production in my herd. So I have to keep on feeding, giving water and medicine to, and impregnating this new calf until I find out what this new calf's milk production is. But I don't want to risk that this calf isn't a good milk producer, so I'm also going to keep on taking care of and impregnating my original cow so that I can get more calves that I also have to look after in the same way until I get some calves that I know are also high milk producers and I can kind of keep the lineage going. Genetic engineering allows us to speed up processes like these so we get animals and crops that we want right out of the gate rather than breeding for traits that we want over many generations. So in nature, many plants have evolved defenses against bugs, including making cyanide, ricin, caffeine, nicotine, tannic acid, which is present in wine. But bugs evolve over time to counter them. So creating genetically modified organisms allows us to bypass using potentially harmful chemicals like pesticides in favor of a protein that's naturally available that doesn't affect us. We can make transgenic plants in which a gene from one organism is engineered into another organism. There's a bacteria called Bt that makes a protein called incidentally Bt toxin that kills bugs but is harmless to us.
Since bugs aren't used to this protein, it's effective to protect plants from bugs. In the case of the peanut plant on the left, the plant on the bottom has the Bt toxin gene incorporated into it, and you can see it's doing fantastically with the larva in its plate. Whereas the plant on the top doesn't have the Bt toxin gene incorporated into it, and it's not doing quite as fantastically. So, on the right over here, we see a normal papaya. However, papayas are commonly infected by viruses. In Hawaii, there was a virus that was threatening to kill all the papayas. By adding a vaccine gene to the papayas through genetic engineering, we were able to save the population of papayas and the farmers' livelihoods. So, in the center, we have a picture of Roundup, which many people know. And Roundup is the weed killer. A protein can be added to our crops that lets them kind of shrug off the weed killer. So rather than the weed killer killing everything, all the plants we want and the plants we don't want, we're able to save the crops that we do want. However, the weeds will still be killed. This allows farmers to more easily get rid of weeds in their fields without risking damage to their crops. So the usefulness of GMOs is definitely caught on. The chart over here, as you see, is the percentage of acres for each of the crops, that is, the genetically modified organism versus the original wild-type organism, over the years from 1996 to 2017. And these have all, for these three species of plant that we're seeing here, this is caught on dramatically. We're seeing here soybeans, cotton, and corn with different kinds of resistance genes added to them. So almost 100% of soybeans now are genetically modified organisms, and well over 75% of corn use is a genetically modified organism. The majority of these changes aren't to the juiciness or the edibility of the plant, but these are related to pest resistance and weed killing. So if you take a look at the list over on the far side of the screen, these are plants that we commonly genetically modify, Max and I, but scientists. So it's estimated that over 100 billion animals were fed with GMOs in the 2000s, although most of these were chickens. And chickens are a very large population of animal compared to other animals. So just how different are genetically modified organisms? Many people find genetically modified organisms concerning because they're different, but just how different are they? Scientists carefully scrutinize DNA from genetically modified organisms to avoid any changes to the DNA other than those changes that we're looking to produce. Mutations happen in both genetically modified organisms and normal organisms when they're bred. However, genetically modified organisms are closely monitored for these changes, while normal plants aren't as carefully observed. I want to direct you to a quote from the journal Plant Physiology that says that there is no evidence that a random genetic change in a crop has ever resulted in a novel safety issue, even when new genes were created. While it is possible to introduce a mutation in a genetically modified organism that could lead to something bad, such as an increase in something that might cause allergies, genetically modified organisms are very carefully tested for these mutations and for any potential effects of a mutation. So nothing with a mutation like this would ever reach the market, but exactly who decides whether a genetically modified organism is safe?
So you make some kind of GMO that you think is the best ever, you have to hand it over to the EPA and the FDA. They do a whole long battery of tests similar to new drug development. We've heard in the news that the drug is something that takes years to come out. The same is true for GMOs. They test for long-term and short-term, if anything in it is toxic. If there's any long-term or short-term changes in how healthy it is for us, how it is, and when it's planted, it's likely or able to spread its genes into other plants like that. Once it's gone through all of their labs, they then hand it over to APHIS, your favorite government agency. They're part of the USDA. And their responsibilities are for checking for invasive species, they check all farms, they look at health threats in agriculture. Anytime you've had a problem importing or exporting food on a plane or something, it's their regulations that are talking about that. They also check on the welfare. And they've even tested if the DNA from any organism, even like a GMO, can pass through our digestive system and into the environment. But no, But if anyone wants to grow a GMO, they need a permit from them. And they'll often have requirements for use, like having a minimum separation distance in the field compared to separated away from other plants and things like that, just to make sure that everything is safe. And this consideration of the environment is something else that we need to think about as we move forward with different kinds of GMOs. How can we make sure that GMOs are safe for the environment? The policy that we constantly advise and talked about, more than just scientists, but also how can we keep the environment safe with the GMOs? Farming the earth, as we mentioned, takes a lot of resources. That fuel and chemical, anytime you don't have a organic or natural fertilizer, this bag of like fertilizer crystals that came from mining in the ground. A lot of herbicides have to be created out of oil. So the risks in agriculture go beyond just the things that we're growing. If we're to feed 3 billion more people in the next 30 years, we need to think about how to do that sustainably. And there are many ideas, so many are still in labs, still being tested, they haven't even gone through a lot of those processes before they get released. So those new research ideas ask how can we make GMOs that can help make sure both us and our neighbors and the earth are healthy. So here are some different ideas we've seen. Is this herbicides, you can plant crops a lot closer together. Old farms have really big separation between all of the plants, so you can spray these quite carefully, but now you can use less farmland to grow this. Nitrogen is a really common fertilizer. It's dug out of the ground, and you can use fewer those kind of chemicals if plants could learn how to take nitrogen straight out of the air, which pea plants can do. So they want to look at bees and see if we can get that one ability, those couple proteins, into something else. So just fertilizer. Climate change is a huge issue that will become worse and worse, and a lot of poorer nations, a lot of third world countries will be hit the hardest because they have fewer resources to really manage a lot of these lands. And GMOs might be able to, for instance, resist drought or if it's flooding too much. All these things that would kill an entire year's worth of plants might have GMOs shrug it off, and you could save populations. And this is a picture of the Brazilian rainforest, a satellite image. Many miles across the satellite image, and you can see all of the Brazilian rainforest that needs to be cut down in order to make more and more farmland. A lot of farmland goes to grains, to meat. So there's one more point about GMOs that's an important issue that we need to solve, and Ali and I are not economists, but we just want to bring it up as an important discussion that a lot of people have different kinds of solutions they're talking about. So these solutions are directly competing, and it's a huge debate. We don't have the answer. We just want you to think about these things, make your own ideas, and discuss them. And any kind of that kind of debate can help solve some problems. And this is, well, life comes from nature. If you find that wild corn, use it. That's nice. It's then studied by scientists like Ali and I, and we're funded by your taxes. Thank you very much. And it's published freely because of that. The results belong to you too. Since they're freely given, the companies can take them, and they're funded by investors who promise a product, and often those GMOs, those seeds that then go to FDA and other agencies are patented so that they can sell them and get back the profit that they invested. If those GMOs turn out to be the one thing that can save the world, that gives the company a lot of power. And a lot of people, I'm sure, since that hasn't exactly totally happened, but sometimes it's little cases, people want to look at that problem and see how can we make sure that this doesn't become a big issue. One example of making sure that GMO crops can't spread their seeds anywhere we don't want them to, besides just separation zones, is making plants that produce seeds that don't give any, where the next generation can't give any crops. They're called terminator seeds because they're the very final round. And so if you do that, you can be sure when the corn blooms and all the pollen isn't going to make a new crop, but then the farmer has to go back to that company every year to buy more seeds. And they use that at that harvest, they can't put new seeds, so that's not a business. On the other hand, if they could just keep all this seed, all they had to sell it once, and then everyone got to use it, maybe no one would ever make GMOs that could solve these problems in the first So there are a lot of these different issues and ideas, and we don't have the answers, but we want to put them up here to show there is a lot of debate about this. And it can be hard to untangle in debates of GMOs what is about how we're using them and what is about what they are. And a lot of times those things are also intricately interconnected.
Alrighty, so now we're going to talk about the elephant in the room, which is, of course, whether humans might be genetically engineered at some point in the future. Early in the talk, I mentioned Huntington's disease was caused by a single mutation in a gene. It actually goes a little bit further than that. Unlike bacteria that have one copy of their DNA, humans have two copies of their DNA, one from their mother and one from their father. This means that you have two copies of the Huntington gene that, if mutated, leads to Huntington's disease. However, you only need to have one bad copy of the gene to get the disease, which is bad enough. However, it is completely random which copy of your Huntington gene you actually pass on to your kids. So, if you have Huntington's disease and you have one bad copy of the Huntington gene, as the light blue man over there has, he has one light blue bad copy and one dark blue good copy. Half of his children are going to have Huntington's disease randomly because half of his children will randomly get his bad copy of the Huntington gene. So, genetic engineering actually gives us a way to avoid a situation like this. Like with the previous example with making green pigs, we can make sure that a child has two normal copies of the Huntington gene when they're only a single cell. That way, a person with Huntington's disease who still wants to have children but wants to make sure that his children don't have the disease can ensure that their kids actually don't have the illness. However, this is not being done by doctors right now. Treating Huntington's disease is a clinical application in which only a single mutation causes the disease. There are other traits like height that not only do tons of genes contribute to, but there are a lot of other factors also involved in these, like what you eat when you're a child and where you grow up. That also have an effect. Things like height are not being changed, both because they are too complicated to change, because they have a large genetic aspect to them, and they also have a large environmental aspect to them. But because also changing them isn't a black and white issue. The decision to engineer these traits lies with everyone, not just scientists. And as you can see, our lab has many people.
So, as I said, this interest not only for decisions, not just in scientists' hands, but these decisions, but also the interest in things like biotech. They can be everyone. So, more and more we see biotech kits and ideas and opportunities to learn getting cheaper and more accessible for everyone. Here's an example of a crowdsourced startup. It's not just the huge corporations that can try to attempt to make something interesting. They wanted to make the glowing plant as a replacement for streetlights. They said if we can make a glowing tree like an avatar, we won't have to pay all this electricity for streetlights, which I believe lighting takes about 10% of our entire electricity in the country. About that much. They wanted just $500,000. They got about $500,000. It eventually failed because it can be hard to predict how to fix these things, how to make these things. And once investors came in, they got pretty difficult. But they tried. There are school kits for all kinds of the similar techniques you saw about the bacteria, about copying DNA. Here is a company called Mini PCR. Everything you see in the photo. That middle box, the brown bar, is what Ali referred to in copying DNA. It's a thermocycling, something called PCR. That on the left is a machine for separating out different pieces of DNA, so you can pick the one you want. And all of that you can buy for about $1,000. So outside maybe most individual ranges for something obviously, but certainly for teachers or something like that, you can do actual these layers of DNA in the classroom and learn about it. There are kits too. This one was a kit to extract your own cell and combine it with different genes for fun, to make them glow, to make them have a red pigment, to make them do fragrances, to make them have enzymes. There's a little kit at home for that kind of fun for $180. And you have, and other things aren't necessarily directly engineering techniques that demonstrate how a lot of these technologies are cheaper. 23andMe and other services like that, sequence, that means reads a lot of the sequences across your DNA for different clues about your ancestry. They don't read all the DNA, but they read certain important pieces. And that is sold for between $100 and $200. And that demonstrates how something like this 10 years ago could have been tens of thousands. And the price of creating new pieces of DNA, the price of reading DNA is plummeting really, really fast. It's changing faster than the speed of computing. So GMOs hold a lot of promise, but as I wrote, they can't be a replacement for being responsible to ourselves and to each other. Maybe we could make the most healthy vegetable in the world, but sometimes I still just like go take a donut instead. And sometimes we make a really good farm, but there's other incentives that are difficult. So we have to be both socially responsible and scientifically responsible. But how we make all those decisions, what regulations and policies we decide, depends on more than just scientists, more than just alumni, depends on all of you and all of your voices, senators reading up about the issues, and the more people talk about them, better decisions can make together to make sure that not only the 3 billion new people, but 7 billion that are on now have a good life and a good planet, and sharing that. Thank you so much for spending your time with us. That will be your questions.